Why don't we get started? Because I want to be mindful of Sarah's and Farzane's time, which yeah. So let's get started, and then it's recorded so people can watch it. So good morning. So we'll as I mentioned every time, this is recorded, so we'll post it on YouTube. So please let us know if you have any concern there. So I have a very brief introductions. The first thing I wanted to mention is that the goal of the experiment that we are laying out is to compare oddballs across different session types. And so in the archive paper that we put together, we highlight that the mice that we are planning to record at the Allen Institute are meant to go through 4 different types of session oddballs. Recorded twice in the 2-photon rigs and once on the NeuroPixels platform. And one of this type is a jitter context. This came about as we were laying out the experimental design. There were 2 projects that attracted a lot of interest across the community. And so one was this comparison of oddball and another was the jitter context. And so as we finalized the experiment, we decided to incorporate both of this experimental design into one. And what we would like to discuss today is one of this jitter context. We are interested in running these experiments at UCL and Georgia Tech in parallel to the Allen Institute. So the intent here is to compare responses from experiment run at the Allen Institute and others in other labs that are potentially pushing more variations so that we can benefit from each other's work. And so today we want to discuss that experimental design, and I was hoping that Sarah would lead the first 10 or 15 minutes discussion.
Okay, sounds good. So yeah, hello everyone. I'm Sarah. I'm based at UCL, and what I'm going to show you in a second is basically work that Farzaneh and I have been coming up based on an interest in how the visual system could form temporal predictions. And so I think what's important to highlight is that most of the mismatches that we discussed until now, I would say, are probably feature-based mismatches in a sense that the orientation of a stimulus might change. It might be halted, so the temporal frequency might change. But at the same time, the standard oddball paradigm has lots of rhythmicity. On the one hand, there's rhythmicity between visual stimuli that Farzaneh will talk about, I think. And on the other hand, the stimulus duration is also standardized and repeated right over and over. And so the experiments that we were interested in trying to do focus on whether the visual system can also form temporal predictions. And we divided it in 2 kinds of assays. So on the one hand, there's the duration of the stimulus that I'm going to show you in a second, the bonsai code that we developed for this, which is just a prototype. And then the other aspect is basically the duration of the gray screen, which in bonsai is called the delay between the visual stimuli, which is another parameter that for now we think of as modulating separately, but there's many combinations one can think of. And so in the first proxy, what we did is we basically modified the standard oddball paradigm to one where the orientation is held constant, but the only thing that's varied is the duration of the visual stimulus. And so I can show you how that looks like in a second to give you a bit of a sense of what this perceptually looks like. So I just give you in a nutshell an overview. So you might have looked at this code before for the standard oddball paradigm. So this is exactly the same template. The only thing that's been varied is the grading specification, which is specifically the duration parameter. And for the example today, I put in a bunch of values that are within the range that we have been discussing informally, but we haven't fully committed yet to the final set of parameters. So the default duration that we use is basically the 343 milliseconds of stimulus duration and gray screen duration that's repeated over and over again. And in this case, the oddball basically is the duration of the stimulus is varied while the gray screen period is held constant. And the examples that I use are basically imagine like a half a second stimulus duration, or basically half of what we use as a default, which is the 170 millisecond, and then some very extreme situation where we have up to 2.5 seconds of stimulus duration. And one thing I think we should discuss is kind of what's the range that we want to use and how we want to parameterize this, because we want to maximize the number of stimulus repetitions. And at the same time, we want to have a kind of a clever division of the jitter that we actually use to maximally be able to predict or basically detect, right, prediction errors at the level of timing. And so when you stimulate this, you get something like that, where this is probably the 343 milliseconds. And then within it, occasionally we have a longer or a shorter one. And the same as with the standard oddball, we can control the ratio of the standards versus the oddballs the way we like, and repeat this n times, and then ultimately follow with receptive grid mapping, which is the code that Jerome basically implemented. And so the question now is, I think at the community level, what are the durations that we want to focus on? Our current stimulus duration itself is already quite short, I would say. So one question is, do we want to make it any shorter? And the other question is, for the longer durations, what are the stimulus durations that of interest to probe to honestly elicit maximal temporal mismatches? So right now, I think the highest number I put in is 2.5 seconds, which is derived from some of Fazene's work that I think used up to 2 seconds for varying the gray screen duration. And the shortest duration, I think you used is 0.5 seconds, but that's very close to the actual standard in terms of the timing that we use in this paradigm. And so I think we should kind of discuss what is a range that we think is suitable, and also how many steps within the range do we actually want to use.
Can I ask a question real quick?
Sure.
Is there any behavioral data or psychophysical data in particular in rodents about time perception? So maybe that can guide us. So the reason that I'm asking is I am aware of some data in humans, but also in other primates to show that while we are talking about time in these absolute terms and linear terms, that is not our perception of time. Mm-hmm. So maybe we might have to factor that in.
Mm-hmm. I think in the rodent literature, there's not much known in terms of, let's say, passive temporal predictions. I think most of what's known in terms of estimating time comes from actively using a motor command to infer the rhythmicity of the action to get a certain reward. Like Joseph Patton, for instance, has done work on that. And so it's not fully clear to what extent the animal in a passive exposure state actually can form temporal predictions. But there's some evidence from some of Fazene's work, for instance, in the past with imaging, that during a mission she found that this VIP population of neurons can ramp up, and so clearly there's at the level of, I would say, half a second, the system can form some temporal anticipation of visual stimuli. But that's now not in the case of the stimulus duration. That's the delay aspect basically that she focused on.
A second question that I have with that is, if we put our neurophysiology hat on, we know that neuroresponses have a dynamics to it, right? So they ramp up, for example, then they kind of adapt, and we get an off response or undershoot afterwards. So I think we might have to be careful, especially if we use brief timing, that what we run into, that we just start to look at these temporal dynamics, right? So we might now mistake an off response as something that wasn't, or we cut an off response short. So that might be another thing to think about.
Yeah, I agree. I think our current stimulus is actually already quite short anyway. And so it's just a bit strange to me to only probe longer durations. My intuition tells me to actually elongate the standard duration as well. Also partially because of the direction tuning. I don't think at the temporal frequency we use, we get a lot of directional tuning because the stimulus is cut off so early on already.
So let me be more concrete. Let's say we have a longer stimulus, right? And so that's our longer oddball or something like that, or maybe also a longer break. Don't we expect, just by the basic properties of neurons. That's if we have a stimulus on for longer, we get more adaptation and we might get a different off response. So how can we guard ourselves from low-level effects causing trivial results?
I mean, I think we can fit, right, the decay of the response to the standard because we have plenty of them. So I think we can actually characterize that, let's say that within stimulus adaptation really well, I would argue. And so in that sense,
sorry to jump in, but could we have controls, for example, a control block where, let's say, we deal with different stimulus durations or gray screen durations? Could we have a control block where we show them repeatedly, and then we can control what if they are an oddball? So then we don't fall for, here's just something that's briefer or shorter. Look, the neurons respond different. Well, we know that. Neurons respond differently to different durations. But we want to know in context, right? We want to know that they respond differently. This was a brief stimulus or short stimulus, brief, break, long break, but that's trivial that would cause different responses in the brain. We want to know that there's an additional different response when those are unexpected. Does that make sense?
Yeah, I agree. I just thought that if we imagine you look at the PSTH over time, you could still compare at each individual moment, right? From the onset of the visual stimulus, the response is not necessarily only the off response that you're interested in when you terminate the stimulus early or late. I thought the control stimulus could actually be just a total scramble of all the durations instead of having something overrepresented.
That'd be perfect. So my concern is, for example, that if you have a longer break, a longer gray screen, you might get a bigger response just because the neurons had more time to recover and are less adapted. So I'm just thinking like a reviewer would if we submit the paper, right? So I think I would say you need a control where you can show me that with an equally long gray screen and I put on a stimulus, but that one is expected, let's say, then you do not get this big response.
Can I ask what we expect? Oh, further, maybe you want.
I can also wait. I was just going to say that I think one thing that would be very helpful is if I share the poster we have presented in a few recent conferences on this same topic, because some of the valid concerns that Alex is raising, we have in our poster. And I think that would be just a very good way
do you want to do that now, or you
I can
prefer?
do it, yeah. And I think, Alex, your concerns are very valid. We have thought about some of those carefully, but I'm pretty sure you have some points also that we may have forgotten. It would be great if you could also write them down, because I'm not sure I followed everything like you spoken like this, and we make sure that we go through this. Okay, so let me share this.
So why do you put it up? Just to reiterate, what I'm advocating for is that if we want to test the unpredictability per se, the ideal contrast when we subtract responses is something where we have the physically same stimulus but predictability, right? So if there's one thing we're interested in and it's unpredictability, the only thing we should vary is not timing and other things, but just the predictability. That's the ideal comparison.
Yeah. Okay, so I'm going to talk about the main points of this poster that we've made. So this is the work that I started when I started my lab 2 years ago, and the entire motivation was to get to the bottom of these omission responses that when I was at the Allen, we were observing in VIP neurons. So the title is a little bit on the cell inside, but it's really the thing because we wanted to see if what we see in these VIP neurons are true prediction error responses. And it seems like what we are finding is that no, they are more about time encoding. So let me tell you about the experimental design. So it's a whole field visual stimuli, and we use BPOD for all of this. Okay, visual and auditory simultaneously. The recordings that I'm showing here are PPC, but we also have from V1, so I can also prepare a presentation of those. And this is VIP and excitatory, but also we have SST, so I can do those on a different session. So this poster is all PPC. So as you see, GCAM8F was injected in VIP-TD tomato mice, and in this way we get simultaneously excitatory and VIP neurons, and this is the thing in PPC. I think would be very helpful to see this. So we did lots of different ways of doing the sessions, and eventually we came up with this very specific paradigm and sequence of sessions. We first start with random sessions. This is a bit confusing, so let me explain what this is about. This is the entire session, and each trial, let's call it a stimulus, an interstimulus interval is a trial, right? So I'm sorry, I feel like I skipped something. So we have these visual stimuli that are 200 millisecond long, and then a gray screen, again, a visual stimulus, okay? And mice are head fixed, and they are also body restricted. So this can make a big difference from the anecdotal things that I've heard around, whether mice are running or not. So our mice are body restrained, which is different from Allen, right? At Allen Jerome, we have them on the disc. Correct. So this is just something to keep in mind. Okay, for in random sessions, the colors here are the 4 different grading orientations, okay? And then the y-axis is showing the interstimulus interval followed by that grading. Because it's very much squished, so it may seem that we have multiple on one X point. It's not true. On one X point we have one, then the other, so it looks like that. The main thing you want to see here is that interstimulus interval varies randomly between 500 and 2,500. And then the orientations also are random. So it's a true random session. We start a naive mouse that has never been in our setup on this, and we give 5 sessions of this. What do we want to assess here? We want to see, because mouse should not be, at least in that very first random session, should not be able to form any prediction whatsoever. Everything is random. With their feature, but our focus, everything in this poster is about the interval. So we started here to see whether we see any kind of ramping, like prediction error, anything like that. Then after that, we go to these structured sessions. And please interrupt me if anything is unclear. We start the session with like a few minutes of random, so it serves as a good control. And then we go to these blocks, short and long. By short, we mean that these images are all at one interval fixed 1 second, and then we switch to long fixed interval 2 seconds. These images are about 200 per block, and within each block we have these mini blocks which are defined by the grading orientation. If you can see, like we have about 20, then 20, 20. So within each block of this fixed interval, we have mini blocks of the other one. The other key feature of this is that one is that it allows us to study, now that we allowed the mouse to have predictions of the interval and then violate them by switching to a different block, and update those predictions. So that's one key thing we wanted to address here. The other is that within each of these blocks, we introduced random oddballs. By oddball here, I mean ISI, interval oddball. I don't mean the oddball in terms of the grading orientation. The orientation, we kept it the same as whatever mini block it was there. But you see, like it's a long block, and all of a sudden we introduce one short interval, and vice versa. This is a short block, and we introduce one long interval. So this design allows us to study emergence of potential predictions within each block, violation and updating. Also within each block, we can see the response to these oddballs. Then after these short, long sessions, we do like 5 of those, 10 of these, and then we do these fixed jitter, what we call it fixed jitter. And this is focus on addressing predictions. This is focus on addressing or studying prediction errors. What do we do here? Again, we start with this random block at the beginning, and then we start blocks that are fixed like this, and this is 1500. And jitter is just like random, going from 500 to 2500. The key is that in both fixed and jitter, we have these oddballs. The oddballs for jitter are just the extremes of this distribution, so they are kind of expected for the mouse, and this is also a flat, uniform distribution. But for jitter, these should be very much surprise events. That was our guess. So we thought this design is the best to address prediction errors. This is for predictions, and this is to see if there is anything there. Because even these, I was concerned that during these, the mouse has already formed a prior of the world because they have seen this for so many days. So this is the experimental design, and all of these take about 1 month. We have 5, 15, 15, 10, something like that. But we try not to switch back or anything. Okay? Everything clear? And you can see
Makes
very
sense.
well the range of intervals. So we stick between 500 to 2500. Short long is 1 second, 2 second, and fixed is 1500. And gradient orient duration is 200 millisecond. We had a lot of difficulty, even though we were using GCAM 8F, with anything below 500 millisecond, and that's why we avoided it, because you'd see the signals. Okay, so this is the thing. Now, what did we find? So, okay.
Can I jump? Can I
Please.
quick question? So I understand the 2 second trials, or even more than 2 seconds. That makes a lot of sense with it's calcium imaging, right?
Correct. 2
Which is
photon,
slow. But
Yeah.
since we have ASAP or SLAP, and we have neurophysiology, which is much faster, I wonder if we can speed up. And the reason that I'm saying that is that if you want to repeat the same experiment, let's say in a primate, 2 seconds is too long, probably. So they struggle to
I think
fixate
I'm going
for
to write down all of these comments so we can come to them. So one is that given SLAP, but we do also have 2P, right? So we should do something that can accommodate our 2P experiments. But given SLAP and if is perhaps how do I want to say it? Reconsider the intervals.
Yeah, I think it's what
Faster.
Sarah initiated as well, to talk about timing, right? So maybe 750 milliseconds instead of 1 hour might work, or something like that.
Sure.
1 hour, sorry, 1
I
second.
should say that we did, what I'm showing here is really after 1 year of trying a lot of parameters. So for 2 photon, these were the best. But I started with much shorter because I also wanted to look at this in better or faster time resolution. But I will write all of these down. Sure.
Can I ask you, these are passively viewing mice, correct?
Correct.
Okay. And because I see you have a lot of plots, and it could take a while. I
No,
feel like
I
it
was
might
going
be
to jump to the main one. I was not going to go through everything. Yeah.
Before you discuss analysis of neuronal data, have you looked into mouse responses in terms of like pupil or movements?
So we do have high-speed video recordings, and we have the pupil data ready. They are doing the summary yet, so I don't have a figure to show for that, but we have it. Yeah.
But because I think Sarah's main point was being sure that the mouse has learned the sequence. And so I'm just curious how you iterated the design so that the mouse learn the blocks quickly, or like what was your approach?
I mean, if I show you this, you'll see that they learn within a trial or so. It's so fast. So fast. So what I'm going to, I'll jump to the, but one thing, Jerome, I want to make sure I follow you. So how is learning this fast is related to your question on pupil, or were they two separate topics?
Well, I think a large number of passively viewing paper that are doing these designs are looking at pupil diameter and seeing that the mouse react to the oddball.
Yeah.
So as a sense that they perceive it, and so that's a proxy we have among other things. But so I'm just curious if you used it or if you're
yes. No,
jumped
that
directly in the neural.
No, that for sure I understood, but I wasn't sure how that's related to the learning the sequence thing that you said, or were they two just separate comments?
I mean, if the mouse doesn't react to the oddball, it's probably a proxy for absence of, like, not enough trials for learning. That's one way to think about it.
Right. Okay. Okay. Let's keep that in mind. So I'm going to jump to the main figure. So for me, if these are, and please tell me if my interpretation is wrong here or my assumption, that this is the gold standard experiment. If the response to these oddballs here and here are not any different, then we are not looking at prediction errors. It just can't be. Because yes, their means are the same. This is way more certain than this. So the responses to these oddballs should be a lot sharper than this. And then and again, I didn't tell you that we're doing more on this to make sure we're not missing anything, but whether excitatory or VIP. And we have clustered our neurons into 3 major groups, the ones that ramp, and the ones that are stimulus driven, and the ones that are stimulus driven but with a delay. So what you're seeing here in blue is the response in the jittered block, and black is the response in the fixed block. This is this long oddball here, this 2500. That's, and then the yellow is the expected time. They expected according to the mean of this distribution, which is 1500. That is the expected time. So grading happens, expected time is 1500, but this is a lot more expected for the black than for the blue. But we see that this is a neuron that ramps slowly after a stimulus, and then whether fixed or jittered block, if the stimulus doesn't happen at the expected time, it does the same thing. It was not like the case that black would be higher because it's a bigger violation. So we have seen this on many mice, many sessions. These just superimpose entirely. We are doing additional analysis now to look more carefully at the variability to see that maybe there is some variability, trial to trial variability that is larger for the fixed blocks than jittered. I would be very surprised if you find anything because error bars don't look to be different, but I will let you know once the analysis is done. For the stimulus-driven neurons, these after the unexpected removal or omission, neither of them do anything, and same with the delay. So this is the thing that we had at the Allen Institute, that we see these ramping neurons, that they kept ramping, and my own interpretation at least was that this might be some prediction error, but now with this fixed and jittered block and them being superimposed, my current interpretation is that this is some interval tracking response and not a prediction error. So I just want to see what everyone thinks about this, because that's the key finding here. Everything else is more getting into the how it adapts across blocks and such, and I can do it at a different time.
I don't want to dominate the discussion, but I have a few quick questions. So the first one is the blue and black responses. Physically, we're looking at the same stimulation, right? So there's it's the same sequence, and then there's an omission in both these cases. So there's no physical difference. Is that right?
So there, this box, this flash, and this flash are the same between blue and black, right? But this one is only for black, because for blue it's jittered, and you can see that black is showing this nice thing, drops down, but blue doesn't have it. But that's because blue is jittered here. This, and this are the same. My student should just remove that. So these 3 are the same between blue and black. And the key is that this.
Nice.
Yeah.
So the only concern here is proving a negative, right? So if you don't see any difference anywhere, how do you know that no prediction, no internal model was formed, and hence no predictions occurred?
There is another one that can be helpful, and that's our random sessions. So this is the very first random session, the very first time mouse sees this. And then what you see here is these beautiful, and this is the last random session. They just do these beautiful ramps, like the stimulus comes, it goes down here. This is the cluster of ramping, not the cluster of stimulus driven is here. I will go there. So what I'm trying to say is that we observe these ramps on the very first exposure of mice to flashes, and it seems, again, this is another indication to me that these are interval tracking neurons, and it's not about the emerging predictions, because if it was predictions, how come we're getting these on the very first thing, the very first session? And the point that you were raising, which was about the longer the interval, the larger the response, you're totally right, and you can see it in our stimulus-driven cluster, that this is larger than that. So yes, there is that. Whether it's it could be a way of encoding the interval in the magnitude of the response, or it could be trivial because there is some recovery, or it could be the dynamics of the dye, the GCAM, that may be the longer we wait its response. So what is underlying this, I can't tell, but and you can see it in our inhibitory neurons nicely, that as the interval increases, the magnitude of the response goes up. So these are things. So this plus that tells me that these are not predictions. And then when we get to our short, long sessions, we wanted to see then, if these are interval tracking, okay, how do they adapt as we move from short to long? It's very rapid within a ISI or two. Again, we are improving these figures. I know they're a bit confusing, but this is the last short ISI, and this is the first long. Yeah, this is more confusing than helpful. We have to improve that, but maybe you can see it here.
Presently?
Yeah.
I think Sarah has a couple questions in the chat. Do the cells have a limit for tracking the interval?
Okay, we've extended it all the way to 2500. I also wonder about that, and you can see it, that it goes. I'd like to know the answer. I guess, yeah, up to 2500 are doing it.
Yeah. And I guess I have a related question. So you show this ramping that emerge, and how many trials does it? Is it on the first trial, or that you present
yes,
the
that's
block
the
start,
one
or how many trials does it take to emerge?
Yes, and that's exactly the purpose of this heat map, is to show that, except that it's made in the most confusing way possible. But this it takes one or two in one interval pretty much. So let's see it here. See, this is short, okay, and this is the very first long. So this is the ramping cluster, right? So the ramping cluster keeps doing that, okay, and then it has a big drop because it managed to go it saw this big interval here, and then do you see this? It just keeps like it took really just a single one here, and then when we go from long to short, so it does that. This is a ramping cluster, so it doesn't care about that. It's that, and then it's really one trial. I think I can show this better. Let me. This is the, yep, okay, I found it. See, this is the continued ramp that we see here, okay?
Yeah.
And then it's the next trial. You can see it. It's literally one trial, one ISI that they do this.
Yeah, so would it be fair to say that the mouse cannot learn it yet? I mean, like you might be looking at a steam off response, and because is the mouse actually perceiving the durations just after one trial or the repeat?
I think it's a very passive process. They're tracking this, and then as soon as it changes, now they adapt. They do this a new one. So here, like in our random one, also, you can see that this tracking continues if it is the stimulus driven, depending on how fast.
I just step back a little bit, because I think the intent of the design is to create a number of complementary oddball contexts so that individual labs can go on to dive deeper into the different contexts and relate it to others. So in my mind, personally, we achieve success if we collect a data set that enrich your explorations and allow to bridge an oddball context that is related to changing orientation with the timing so that people can uh, think about the different types of predictions that are engaged and then how they are done. Um, so I might argue that in terms of experimental design, what we want to favor here is a set of parameters that connect between your lab's work, yours and Sarah's work, and then the others. And so in what would be your perspective given that? Because I think Sarah asked about the durations and
So for the durations, I was thinking of like being close to these, the ones that I showed. Alex had the comment that maybe we want to do something that also is better suitable for SLAP or IFIS. If I don't want to be worried about SLAP or IFIS, I would go with similar intervals, which or durations as
because your
ours.
intervals are like what they're like half a second.
Half a second all the way to 2.5, and our grading duration is 200.
Because our current period is 343 milliseconds.
Yes, it's pretty close. I noticed it, but it's not too far. But we can do it like, and it's a bit lower.
And what is the concern is that it's too short to see the ramp? Is that the concern?
It's too short to see the ramp, and also when the stimulus response before it decays, we get another one. Like I wanted to have a very nice view of the response of the cell, and for that I had to give a good. And by the way, I didn't show the responses to the short oddball ISIs. For those, it was very hard when we had them too short.
But but again, in the 2-photon data we collected before the detection of change, we saw the ramp, right? And then the duration was even shorter, I recall.
We had it at 750 at Allen, if I remember, right? We had 250 stimulus 750, so it was not
right. The gray period is longer. Yeah.
Yeah, it was 250 750. If I
But here you
remember.
have 343 times 2, so it's 800 milliseconds.
You're right. So 342. I think it would be helpful if these numbers that Sarah says is written somewhere, because I keep getting confused. Like, what is the grading duration that Sarah has now?
340.
340 millisecond. And what is the minimum or range of well, grading duration is supposed to be variable, right?
Well, the baseline is for 340.
Okay, and what is the range?
I think in terms of oddball.
Yeah, we are going to change it, right? Yes.
So she's asking. Yeah, well, I think she was proposing to use some of the values you were using here. She used the range 0.5 to 2.5 seconds.
0.5, yeah.
For the
exactly.
oddballs, yeah.
For oddballs, and then the grade duration is what?
Is the same, 340.
340 millisecond. So
I think you should see the ramping here.
With 340?
Yeah. I mean, it might be on the short end, but would you agree?
Yes, I think we would see it. It won't be as nice and clear, but we would see it. When we started this, we did 200 millisecond interval. And with that, it was a bit messy.
But remember, personally, you are going to get neuropixel data in addition. So you're going to have high, and then the advantage of that is that you're going to get a whole flurry of subcortical areas that are probably involved here.
Right.
in
I
timing.
mean, for me, it was important because we are doing these same experiments on the cerebellum. And for me, it was important to have the range from sub-second.
Especially the hippocampus that is likely going to influence V1 and provide a little more timing.
So you prefer to have it more grind here?
What do you mean?
I mean, more like detailed, like lower?
I would prefer to have the same baseline as a standard double session, because then you can compare.
What is the standard double session?
343.
Standard double session for the grade duration, you mean?
For the grading and the grade durations.
Yeah,
because in the standard oddball, we are not changing durations.
Yes, Jerome, I'm not too worried about this. This is not too short at all. So from going from 500 to 350, perhaps we won't get some of these very nice ramps, but it's definitely good enough to give us the ramps. I'm not too worried. As I'm saying, when I started, I did 200 millisecond and still I could see ramps, but it was just not clean. So that's why I went. But I think 340 is really not, I don't have anything against it.
Yeah, we can put things in writing and then you can think about it more deeply. But
yeah, I'm writing things here. Yeah. And some of the questions that Alex had, I think were very important. And that would be helpful if Alex, you just write an email to all of us and write your points. I have tried to think very carefully about alternative explanations for these, and I think you were getting at some of those.
There's a recent paper from Bujaki lab that
I noticed it. Yeah, we are doing it in our lab next week. Yeah, I saw that post. I saw their poster.
I must say, I think his result probably are in alignment with yours.
Okay, yeah, that's what I saw in their thing too. Yeah, and we have these for V1 also, but before saying I prefer to have the figures before I talk about it, so I don't make any mistakes.
So okay. I have a meta level question. So if I understand correctly, what you show here is that there is no predictive coding.
Not for time.
So then why should we do all of this again, the whole experiment?
Well, first of all, that's why I'm very curious about the duration, because that's a different one. Second, I still have to, our SST might be showing something, but again, I'm very careful about talking without having the figures in front of me. But yes, I know based on interval PPC V1, we are seeing this. But whether it's going to be different for duration,
So I have an additional. Just a quick, I have an additional concern here that if I were the devil's advocate for predictive coding, I would say there's something about this particular stimulus design that doesn't allow for prediction to form. Or maybe it's if the mice would do it longer or something. I would still try to find some way to say, yes, there would be predictive coding if you would do things differently.
Temporal predictive coding you're talking about, right? We are talking. So as I'm saying, I'm also thinking maybe the cerebellum is doing it. So we are looking at that too. It should be somewhere in the brain is tracking that, right? So we didn't.
How do you know that? How do you know that?
That why the brain would keep track of violations of time?
Yeah. In a rodent.
Why would the brain is it not like a basic computation that we would expect the brain to be able to do to tell if the time is violated?
Well, only if we have behavioral evidence that that computation system does that, right? And so that was my first question, that is not at all obvious. So humans, for example, we are very good with rhythm, but you rarely find this in other animals.
So what we're doing now is that we're doing these same experiments in an active paradigm that mouse actually needs to attend to those to make a decision. And so that was the thing that I said, maybe we don't see it for passive, but maybe it's for active. But yeah, one answer also is that maybe because it's passive, the mouse is not caring to keep track of temporal.
Alex, I might argue against devil's advocate, but we'll, given the recent result from the analysis of Bujaki lab on our data, you probably are more likely to see it in hippocampus, right? Because the slope, I recollect the figure, the slope was very high in terms of responses to the omission in the hippocampus compared to V1. And so, and also, given the literature, there's probably better sense of timing there. And so I feel like then in that particular case, it will mean that it's especially important to record from the hippocampus in those
Jaron,
hardball
what is the
tasks.
time range they are working with in Buzaki paper?
It's 250 and 750 milliseconds with an
Okay,
he's
sub-second.
analyzing the omission, so that's different, right? It's very different. But
Right.
in the omission, the presence of omission is, I think, it's only in the hippocampus we see very strong response locked to the prediction.
Yeah, but they don't have the jitter test, do they?
They don't.
Yeah, so my conclusion after all of these things that we've done is that I'm not going to buy anything about prediction errors unless you do the right controls there, because not every violation is necessarily prediction error. Like change down the certainty of the environment and then show me that it actually decreases, because that's one of the key features defining the prediction error. Yeah.
I mean,
So they have
an
an omission response, but whether it's a prediction error or not.
Hamid, did you have a question?
Oh yeah, I was just quickly going to add that in Monkey's omission response, we see it like widespread in LFP too. And this time scales is like in 501 second, and it's locked to the onset of expected. So in Buzaki paper, it was rodents. They found it significant only in hippocampus, I think. But in primate was like really, it's a really different neural signature as we observe.
And you guys see it in cortex, you're saying?
Yeah, V1, yeah. V2, only in LFP, V1, V2, PF, all of them, but spiking only PF, CFF, as before.
So Hamid, that could still be then the hippocampus, right? So if you measure LFP in V1, that can originate in the hippocampus.
Yeah, we are not recording from hippocampus, but we see a very strong single neurons ramping to it in PF, CFF.
Or it's that, right? But if the LFP, you can measure a correlate of the LFP on the scalp. That doesn't mean they have neurons right under the scalp, right? So these are radio signals, and you can get them from anywhere. So if you see an LFP signal anywhere, that just means that it's somewhere happening, but you don't know where it's happening.
Yeah. Especially because there is not even a single neuron in V1 responding, but there's LFP. That's one example that the origin might be somewhere else, but observation might be everywhere.
So I want to add one more point. We never have seen any neurons that respond purely to the omission. Our responses are this continuation of ramps. So, and I think this is again another sign that these are interval coding response, because if the neuron was silent and then responds when the omission happens, we don't have that thing. Does Buzaki show that for hippocampus, that is it a continuation of ramp or is it actually an omission response? Anyone remembers?
I think it's I think they have both in the paper.
Okay, I will have to also look at that carefully, but we didn't see.
They show the beautiful ramping neurons. They measure the slope of the neurons, like essentially the slope of the ramp, and they measure the slope across all of the brain areas we recorded in neuropixel data. Yeah. And you see that the slope is very high in the hippocampus.
And when you say ramp, you mean, because our ramp starts slowly after the previous stimulus, and then it continues when the expected stimulus is gone. So it's very different if the neuron is silent and then starts respond. We do have ramps that start later.
Yeah, the slope is measured at the omission time.
Okay.
So it's focused on analysis of omission, so it's different.
Yeah.
But I think it's related.
Yeah.
Yeah. Okay.
So just to, again, on the meta level, so why will we do all of this effort if there may not be what we're looking for. So then the answer is Bushaki's lab found something, right? So is that the answer, that we should also find something if we stick close to what he did?
I think so.
I have an answer for that. One is that, one is the different modalities of recording. The other is that we have looked at V1 PPC and cerebellum. And with this, the idea is to look further at other areas, including the hippocampus. Then is that maybe there is a difference between interval and duration. But I agree, if you're talking about V1 PPC interval, that's what we've done for 2 years now, and we have these results. And I would be surprised if we find something else. Unless neuropixels show something, which means then we should just not do image.
Is anybody recording from premotor areas or motor areas with that? Would that be feasible?
So for it is totally feasible for us, and we have windows on the PFC. But yeah, so we've been.
So
yeah.
Sorry, you meant motor cortex?
Right. So because even in M1 you find visual responses, and it's kind of a puzzle, right? So, but I think about animals. Where does rhythmicity matter? And again, there's some evidence that they don't have absolute time perception. They count events. It doesn't matter how spaced out they are. So behavioral data. But where rhythmicity actually matters is, for example, in locomotion. And so you could think that's why, you're already doing the cerebellum. That's one thing that I'd be thinking about as well. So which structures in the brain are actually interested in rhythmicity?
Yeah, the recording plan at the Allen is to record from the motor cortex. Yeah, it's in the archive paper.
So it's also was hinted in the discussion. You might think that auditory oddballs are more prone to this, right? So in the auditory system, absolute timing matters a lot. And so there's some thought that the auditory cortex, even anatomically, is designed differently because time is such an important dimension in audition. Whereas in vision, space is the dominating information. And so maybe if you have auditory oddballs, things might be different also than visual oddballs.
Yeah, although you might argue that for a mouse, perceiving something that has an odd timing is a matter of survival, because they're probably using the vision to screen around quickly, and so if something is not following the expected direction, I would expect to jump away.
If we can show that, then we're golden, right? But we only have to show that what we're looking for exists. Because a skeptic like me could say, you're looking at an animal that's designed to live for a year or two. It doesn't need that. It doesn't do that. So, but if we have pupils or something else, then if we can show the brain does it, we don't know where. And that's what we want to find. That helps us out, because right now it looks like, well, we look, we don't find it. Why do we do it? Then even look even further.
Yeah. We are close to the hour, and so I think the plan in the next few weeks for me is to take the jitter bonsai files and adapt it so that we run at the Allen, and so we need to sort of start thinking about the parameters and things like that. As far as any other experiments that are ongoing that would influence those parameters that we should think about?
No, because if anything, we are just using the same. We are not using parameters in our lab at this point. We are just collecting more data. I also want to just emphasize one more point that I have said, but it's important. We are doing audio visual simultaneously. And this is also another difference with the Allen.
I see. Have you seen differences to Alex's point with audio?
What is that again?
If you present all auditory periods, do you see the same ramping behavior?
Our stimuli are always congruent. So
I see.
Yes, they're always congruent. And the other major difference is that our mice are in the IBL kind of tube. So I think these are two big differences that I can imagine. Well, the audio-visual, I'm not too positive, but the tube can be a thing here.
Okay.
For the hippocampus, it might matter, right? So I can think that the hippocampus is supposed to be in the rodent mostly about spatial navigation. And so then timing might really matter. right? So if you're trying to go through a maze, you kind of have to maybe keep track of time, how much distance you have done for your internal map of where you are. So if they're immobile, then maybe you don't see even in the hippocampus what you're looking for.
Well, yeah, yes.
Okay.
I don't think they're entirely immobile. They still move, but yeah, they are not locomotive. Yeah.
I think we're close to the hour. I think the goal of the meeting was to facilitate this conversation. I think we achieved that. So we can continue. I think we can continue to discuss this on the forum, on the GitHub forum.
I have one last question, real quick, Jerome. So, and it's what you will do. So, is the idea that once you implement it in the next 2 weeks or so, you'll also try it in the SLAP experiment?
We are not planning that. I can discuss with Casper offline and we could see. But yeah, Casper, do you have thoughts there? Or you're on board, or?
I'm interested in doing all of the experiments that are implementable on the current visual observatory rig on there. We just need to schedule it along with other things. But yeah, in principle, interested.
So the reason that I'm saying that is we started analyzing the oddballs, right? And so I think we ran into a shortcoming, which is if we only look at one neuron and we look at oddballs as different orientations, it becomes very difficult to disentangle the orientation response from the actual oddball response. And so if we have a different kind of oddball where we can get around that, then the single neuron experiment might become more interesting. So right now we see that.
I've been advocating for a while of actually simplifying, or in my mind at least, I've been advocating, maybe I haven't been advocating to the group, that we should kind of simplify this experiment so that we get a lot of trials of things when they are expected versus unexpected. It doesn't change the, it doesn't fully address the thing that you're saying right now. But certainly getting the output of the cell, getting the many synapses which represent the population, and then designing the experiment such that we get a lot of focusing measurements on measuring the oddball when it's not the oddball and measuring it when it is the oddball and sort of flipping it, I think would help what you're saying. It's a different discussion, sorry.
I think that might be next week. I propose that we discuss the analysis of SLAP2, Alex. And so I think if you could present what you've done with Sarah and any others that want, I think that would be wonderful. And then we could sort of use this as a lesson learned and then influence the rest of the design. Does that make sense? That's a good proxy. So next week, I would like us to discuss the SLAP2 analysis. There's already some great progress made. And then the following week, I think it would be great, but we discuss the sensory motor context. Okay. Please engage on the forum, on the GitHub forum. I think we should do our best to kickstart this discussion place. There's already quite a lot of interesting discussion there, but I think this is the place where we can gather and continue the conversations in depth with plots and everything. So if you have not done so, please check it out. Okay. Any other questions before we part? Next week we'll probably go back to the normal time. Well, thank you.
Thank you all. Bye.
Thank you.